reductionist approach to the study of learning and memory. So let's see if we can take a single sensory neuron, a single motor neuron, and a single serotonergic neuron and put it in a dish. And we can do that. Sensory neuron on top forms perfectly good connections with the motor neurons, and the serotonergic cells, the modular neurons, form connections onto them. And if you stimulate the modulatory neurons, you'll strengthen the connections between the sensory neuron and the motor neuron. But we don't even need the modulatory neurons. We know they release serotonin. So we can substitute a pipette that simply puffs on serotonin. We now have a learning circuit, a 2-neuron learning circuit in a dish, and we can now puff on serotonin. And we can see what happens. If we puff on serotonin once, we get a transient facilitation of synaptic strength. We're now looking at the amplitude of synaptic strength as a function of time. Lasts minutes. If we now give 5 puffs, brief puffs of serotonin, we get a facilitation that lasts more than 1 day. And now we can ask something very nice. We saw that long-term memory differs from short-term memory in requiring the synthesis of new proteins. Is this represented at the elementary level? Can you see this at the connection between the sensory neuron and the motor neuron? And we can obviously do this very simply. We can apply inhibitors of protein synthesis, and we find that it selectively blocks the long-term without in any way affecting the short-term process. So here we have a situation in which we reduce the learning process, a component of it, a critical component of it, to a monosynaptic connection consisting of literally 2 cells and a puff of pipette. And we can now try to analyze the molecular mechanisms that underlie it. So I'm going to show you a blow-up of the sensory neuron and the motor neuron and the serotonergic modulatory inputs. So sensory neuron is on top, motor neuron is at the bottom. On the left-hand side, you see the tail-activating serotonergic cells. Let's look at the basal condition. In the absence of tail stimulation, we generate an action potential in the sensory neurons. As Tom Jessel pointed out in his lecture, that propagates to the presynaptic terminal. It opens up calcium channels, allows calcium to come into the presynaptic terminal, and calcium is absolutely required for the exocytotic release of transmitter. It allows vesicles to bind and to be released. What happens if we now stimulate the tail? We activate serotonergic cells. These release serotonin. Serotonin is recognized by a serotonergic receptor in the presynaptic sensory neurons. These engage an enzyme called an adenyl cyclase that converts ATP to cyclic AMP. Cyclic AMP is a second messenger. There are lots of different second messengers. They all have the same function. They carry information from the membrane surface to the molecular machinery inside the cell. Cyclic AMP activates a particular molecular machine called the cyclic AMP-dependent protein kinase. That is abbreviated protein kinase A, pKa. Cyclic AMP-dependent protein kinase has 2 subunits, a regulatory subunit and a catalytic subunit. I've indicated the regulatory subunit as a spindle-shaped structure and the catalytic subunit as an oval-shaped structure. The regulatory subunit inhibits the catalytic subunit under normal circumstances. When the level of cyclic AMP rises, the regulatory subunit binds cyclic AMP, undergoes a conformational change, frees the catalytic subunit. It now can move into the presynaptic terminal. It acts on ion channels. It phosphorylates them, alters their properties, allows calcium to come into the presynaptic terminal. So now an action potential has more calcium coming in, more vesicles can bind, more vesicles can be released, and you have enhanced transmitter release. And that is the mechanism of short-term memory for synaptic facilitation. And this is occurring at every single sensory neuron that is involved in the reflex. What about long-term memory? When we give repeated tail shock, we activate the serotonergic cells more. They release more serotonin, increases the level of cyclic AMP more, activates the cyclic AMP-dependent protein kinase more persistently. So now the cyclic AMP-dependent protein kinase can do 2 jobs, not just 1. Number one, it goes to the presynaptic terminal where it enhances transmitter release as it did in the short term. But now it does something very interestingly. The catalytic subunit moves into the nucleus. So one of the reasons you need repeated training is to allow second messenger kinases to move into the nucleus. And there they activate transcription. They act on a transcription factor, and Tom told you what transcription factors are. They're proteins that bind to specific sequences in DNA. This transcription factor is called CREB, cyclic AMP response element binding protein. And it binds to a sequence in DNA called the CRE, cyclic AMP response element. When it becomes phosphorylated by pKa, it can bind to that sequence, activate genes, and these give rise to the growth of new synaptic connections. And it's the growth of new synaptic connections that is the maintained form of memory storage. This growth is what is responsible for the maintained strength of synaptic connections. And if you leave this room remembering Tom's lecture, you will actually have an anatomical change as a result of this. So there are two really interesting things that I want you to think about. The first is that long-term memory differs from short-term memory in involving the expression of genes, altering gene expression. Now, you heard from Tom, and we generally think of genes as being the controllers of behavior, the determinants of behavior, and that is true. They're extremely important. But what is not also appreciated is that genes are also the servants of the environment. They respond to environmental contingencies. So when you interact with each other for a long period of time and affect each other's behavior, you're producing alterations in gene expression. And gene expression is particularly important because it is the gene expression that gives rise to the growth of new synaptic connections. The gene products that are produced are responsible for the growth of new synaptic connections, and they're absolutely required for that. If you block CREB, you don't get a long-term memory. You don't get a growth of new synaptic connections. Now, what are the kinds of proteins that are required here? Well, two of the proteins that are required are neurexin in the presynaptic neuron and neuroligin in the postsynaptic cell. Have we heard about these before? Yes. Tom brought them in as being zippers, important in developmental synapse formation. And these studies show they're also important in the new growth associated with learning. And that suggests the possibility that one of the things that is defective in autism, in which these particular genes mutated in certain individuals, is that they lack the capability to learn certain aspects of social interaction. In aplasia, the growth is really quite dramatic. So this is a sensory neuron. Here's a motor neuron before learning. And here, after long-term sensitization training, you see an outgrowth, a doubling of number of synaptic connections. In the short term, the change is presynaptic. Any long-term process involving growth has to be coordinated. So it's both pre- and postsynaptic. Now, you can argue this is only a snail. Does this apply to you? Does this apply to me? And I want to convince you that this is happening in your brain. This is the motor strip in your cortex. We talked about this yesterday, in which every movement of your body is represented. There's a hand representation. And when I was a medical student, I was taught that the hand representation in my head is fixed. It cannot be changed. And we now know this is not true. That if you simply do what Tom had you do, oppose your thumb to your finger, and you do this all the time, you get better at it. And that is because you're actually changing the size of the representation of the fingers in your brain. And we can show this very nicely in an experiment by Traub, in which he looked at the representation of the left hand involved in fingering the violin. And he looked at that representation in control people who did not play the violin and in string instrument players. And he found that the string instrument players who fingered the violin all the time with their left hand, did the fingering, had a much larger representation than those who don't play a string instrument. And this answers one of the questions that emerged earlier, if you do what your parents told you to do, you start playing early and practice a lot, your representation will be much greater than if you start later on in life. So what does this say? We have reason to believe that you can continue to learn new stuff throughout your whole life, and you can continue to grow new connections throughout life. But the capability to do that varies as a function of age, and it's magical in the early years. This has several implications. It tells you that Michael Jordan is Michael Jordan and Mozart are Mozart, not simply because they have the genes for the genius, which they do, but also because they began to practice their skills very early in life. You have an enormous capability, and this still applies to you at this point, to acquire new information. These are the magical years for growing your brain. Number one. Number two, it means that every single person in this room has a different brain than every other person in this room, if only because you've had different social interactions. Identical twins with identical genes will have somewhat different brains because they've had different kinds of intellectual experiences. So I will go on to consider explicit memory storage with you, but I will stop now to take a few questions from you.